Hello, everyone. A very good evening to one and all. I hope you all are doing good. Myself, I am Dietitian Aisha Rabbani from AgriVision4U, and I'm your mentor for the course to crack the upcoming N-CET exam, which is on 23rd June 2024. So today I'll be discussing about the topic, about certain cycles a little in detail. Okay. So to begin with, let me just share my screen. So these cycles, okay, we have certain metabolisms, like we have the carbohydrate metabolism, we are having the amino acid metabolism, okay, and much more like this. So for this, there are certain procedures or certain cycles that takes place, to break down this into a further simpler product. For example, to the very first one to discuss with, okay, uh, we'll discuss about urea cycle, but for yourself is the important cycles. It is, the urea cycle you're having tricarboxylic acid, you're having glycolysis, you're having pentose phosphate pathway. So to start with urea cycle, it is, the end product, or I can say, end product of the protein metabolism, or I can say it is the amino acid metabolism. So to understand these cycles, you have to first understand what is exactly the process of digestion and absorption of carbohydrates and proteins and certain vitamins and minerals and, all the vital nutrients. How does it exactly get absorbed into the body? Why this cycles are important, and the questions or the type of questions that will be asked, the steps, the essential steps of each and every cycle along with the energetics. You have to be very thorough. To begin with urea cycle, as I've already mentioned, that it is, the end product of the protein metabolism, which is also known as the amino acid metabolism. So the nitrogen atom of the amino acid, it is, getting converted to ammonia. And you all know that this ammonia is very toxic to the body. It is then converted to urea and it is finally detoxified. See, urea, it usually accounts for, 80 to 90% of the nitrogen containing substances that are usually excreted in the urine. So urea is, synthesized in the liver and transported to the kidneys for excretion in the urine. The urea cycle, I can say it is the first metabolic cycle. Okay, it is the first metabolic cycle, and it was elucidated, or you can say it was established or given by Hans Krebs and Kurt Henslett cycle. Okay, Kurt Henslett, he both of these scientists have given this urea cycle in the year 1932. Okay, that is the reason why this urea cycle is, usually known as Krebs-Henslett cycle. Okay. So there will be certain MCQs that urea cycle is also known as. So they'll give you in the option Krebs cycle and Krebs-Henslett cycle. Now many of them get confused between this TC and urea cycle, because the tricarboxylic acid cycle is usually known as the Krebs cycle, but this urea cycle is known as Krebs-Henslett cycle. So either of the question asked, both the students get confused in again both the options. Krebs cycle is different. Krebs-Henslett cycle is different. Okay, so that is how you all have to remember this difference. Okay, I am mentioning it over here so that it doesn't get, like it doesn't create any kind of confusion. So this individual reactions, there are certain steps, okay, which we'll be discussing now. So the individual reactions of these steps were described in more detail by Bratner and Cohen. Okay, Urea, it has two amino groups. One amine group is derived from ammonia, and the other one is usually derived from the aspartate. Carbon atom is supplied by the carbon dioxide. In urea synthesis, it takes in five step, which is a cyclic process, as you can see in the picture also, and it has five distinct enzymes. Okay, the numbering has been given that the carbamyl phosphate synthetase is the first enzyme, and then you are having ornithine, and then you are having arginosuccinate synthase, then you are having arginosuccinate lyase and arginase. These in order you have to remember, like without fail, because these are important. Okay. Um, and the first two enzymes, since this urea cycle is actually divided into two parts. The first part is taken into the mitochondria, okay, while the other is usually localized in the cytosol. So the first two enzymes, that is the carbamyl phosphate synthetase and ornithine carbamyl transferase, these are located, and or you can say these are present in the mitochondria, while the rest three, that is arginosuccinate synthase, arginosuccinate lyase, and arginase. These are located in the cytosol. So there are basic five steps. Okay. Firstly, in this cycle, there is synthesis of carbamyl phosphate. Okay. And then you are having formation of citrulline. And then you are having synthesis of arginosuccinate. And then you are having cleavage of arginosuccinate again. And then the final step is the formation of urea. These are the five essential steps that takes place in the urea cycle. Okay. This carbamyl phosphate synthase, okay, it like in which is present in the mitochondria. It is catalyzing the condensation of the ammonium ions, NH4 plus ions, okay, along with carbon dioxide to form this carbamyl phosphate. This particular step, it consumes 2 ATP and it is irreversible. So, this carbamyl phosphate synthase requires N-acetyl glutamate for its activity. And there is another enzyme, carbamyl phosphate synthase. Like there are two. This carbamyl phosphate synthase 1 and then carbamyl phosphate synthase 2. CPS1, CPS2, like that it is. So the first one, it catalyzes the condensation of ammonium ions, and the other one is involved in the pyrimidine synthesis. Okay, and it is present in the cytosol. So it accepts amino group from the glutamine, and it does not require any kind of N-acetyl glutamate for its activity. I'm talking about the CPS2. Then, with this, citrulline is formed. It is like synthesized from the carbamyl phosphate and ornithine by ornithine carbamyl transferase. Okay, this ornithine is, regenerated and usually used in um, the urea cycle. So its role is comparable to that of oxaloacetate in the citric acid cycle, which we'll be discussing pretty soon. Ornithine and citrulline are basic amino acids, and they are never found in the protein structure due to the lack of codons. Citrulline produced in this reaction is transported to cytosol via transporter system. Okay, and then there is synthesis of arginosuccinate. This arginosuccinate synthase condenses the citrulline with aspartate to produce arginosuccinate, and the second amino group of urea is incorporated in this reaction. This step, it requires ATP, which is cleaved to AMP. I hope you all know what is the full form of AMP, because these full form, whether it is AMP or ADP or anything, it should be on your fingertips. So This AMP, it actually stands for adenosine monophosphate. Okay. And then there are certain steps which we'll be discussing it in more detail. Okay, like how cleavage of arganosuccinate is formed, then finally there's formation of urea, and there are energetics and all. So this synthesis of arganosuccinate, it condenses citrulline with aspartate to produce arganosuccinate. There is second amino group of urea incorporated in this reaction. And this step requires ATP, which is cleaved to this AMP, and then there is a pyrophosphate, usually known as PPI. And the latter is immediately broken down into the inorganic phosphate. Inorganic phosphate is PI. PPI is pyrophosphate, and this is inorganic phosphate. Okay. Cleavage of arganosuccinate, it cleaves to give arginine and fumarate. Arginine is usually the immediate precursor for urea. And fumarate liberated here, it provides a connecting link with TCA cycle and gluconeogenesis usually. If I talk about the final step, that is the formation of urea, it is the fifth and the final enzyme, arginase, that cleaves arginine to yield urea and ornithine, as you can see here. This ornithine is again regenerated, and it enters mitochondria for its reuse in the urea cycle. Arginase is activated by cobalt and manganese ions, CO2 plus and Mn2 plus. I hope you all know this abbreviations. So ornithine and lysine, they compete with arginine, which is actually known as competitive inhibition. Arginase is mostly found in the liver, while the rest of the enzymes, like four of the urea cycle, are present in the other tissues. So for this reason, this arginine synthesis, it may occur to varying degree in many tissues, but only the liver can ultimately produce urea. All right. I hope like it's clearly understood. And the energetics is usually this way, like the overall reaction. Like this it is. So you have to remember. The urea cycle is irreversible. Okay, it consumes 4 ATP. 2 ATP are utilized for the synthesis of carbamyl phosphate, and 1 ATP is converted to AMP and the pyrophosphate to produce arginosuccinate, which is equals to the 2 ATP. So hence 4 ATP are actually consumed in this. Certain things like this you have to remember. Okay, so usually they ask which is the first metabolic cycle, or urea cycle is also known as. Then they can give you a reaction of chain, um, where they involve the enzymes. So you have to arrange it in the order. So remembering it in the order, this picture will help you a lot. Along with the numberings you write or you learn by heart, however you practice, it depends on you. But cycles should be on your fingertips, because majority questions in the biochemistry, they ask from cycles only. Then they go for molecular bases in other things, principles and variations involved in heredity and all such questions they ask, but majorly they ask from the cycles. So I hope that you have understood the urea cycle pretty well. There are other cycles that we'll be discussing it in our live classes at AgriVision4U, okay, which is bringing a crash course, to crack the exam and get in-depth knowledge. So it will be starting pretty soon. So for enrollment and further details, you can contact on the number which is shared on the screen. Thank you so much.